Multiomic analysis is a new emerging field in medicine that allows us to understand what is happening inside the human body. It's not just inside the human body, it also allows us to understand what is happening on the tissue level and what's also happening inside the singular cell, inside just one cell inside this tissue. So to have an idea what multiomic analysis is, imagine a city full of factories. So we have many different factories and they are all producing products. They are working on to make these new products and hopefully to let the economy grow inside the city. So in the last decades, multiomic analysis was able to give us a screenshot of the city, of the economy of the city. So we could see that, okay, everything's working, the economy maybe is not growing. It seems that the factories are not working at full level, but we don't really know which factories are working at high levels, at 100%, which factories are working at 60 or 10%, or maybe not working at all. But we have this idea that, okay, production is working, it seems that the economy is still working, everything is fine. And in the last years, especially during and after the COVID pandemic, the technology in multi-omic analysis has made great leaps forward that allows us to have yet a more detailed understanding. We're now able to understand, we're now able to look inside the cell, inside a single cell, to see, okay, what is happening in the genes, what's being transcribed, which drugs are being metabolites, which drugs are not. So if we go back to the analogy of the city with the factories. In the old days, we had the screenshot, we had a form of a mean description, what is happening inside the city. Okay, 60% production is working, that's it. Now, we are able, or if we took the analogy of the city with the factories, we can see these factories are like tissues inside the body. So the city is the body, and these factories are represents the tissues inside the body. But the difference is that the body does not consist of thousands of factories. It consists of millions of factories, millions of tissues in this example. And using multi-omic analysis allows us to understand not just what is happening inside the factory. It allows us to understand it allows us to understand every detail about every single factory. So we're able to understand what the workers are talking about, what they are eating, where they're spending their time. We're able to understand what is the blueprint inside this factory, what's being communicated, what's being produced, which products are useful, which products are not. And if you take this analogy to the body, we're able, using multiomic analysis, we're able to understand what's happening inside the cell, what is being transcribed inside the cell, which part of these transcriptions are leading to proteins, and which part of these proteins do have a function, or which part of these proteins are being phosphorylized, and what kind of changes are happening to these proteins. So we are able to have very great detailed understanding of every single tissue, every single cell inside the body. This is what multi-omic analysis is about. Another way to understand what multi-omics is all about is by looking at the word. multiomics. So multiomics is consists of two words, multi and omics. So it's a prefix and it's a suffix. Prefix is the multi and the suffix is omics. So everyone knows what multi is, so we will not dive into that. But what about omics? So omics describes the study of the totality of something. I'll repeat it again. So omics describes, or omics is about studying the totality of something, the totality of something. So if you understand what omics is about, now you understand the whole aspects, the whole thing about multiomics. So it's about understanding the totality of something. Now, if we replace multi with something else, let's say genes, for example, so we have something called genomics. So gene from the genes, genomics. So genomics is about understanding the totality of the genes. What is the totality of the genes? Well, it's about understanding the whole DNA of a single human being. So human beings, our genome, our genes, our genome consists of 3.2 billion base pairs. So genomics is about analyzing the whole genome of the body. We can also analyze parts of the genome. This would also be genomics, but some information will be missing. So when we're talking about genomics, we want to understand what our blueprint is about. What are the genes inside of us? What is the DNA? What's the structure of the DNA? Which genes are there? Which genes have been changed or had certain mutations? And using genomics, we are able to find certain mutations inside the genes. For example, deletion. So when part of the genes has been deleted, this is something that this can be related to pathological changes in the body, so syndromes, for example. I just talked about deletion. So when part of the genes has been deleted, this might lead to a disease. One example of this would be cystic fibrosis. So when part of a certain gene is being deleted in the genome, in our DNA, then people with this type of gene mutation will develop a disease called cystic fibrosis. And we're not just looking at deletions, we're looking at many different things, like inserting new nucleotides inside the DNA. We're also looking at inversion when parts of the DNA has been swapped or switched. So inversion is something like reading a phone number not from left to right, but reading it from right to left. It's a totally different phone number. And the same thing might happen if parts of the DNA has been inverted, something called inversion. So in genomics, we are looking at these changes, but we're not just looking at changes in the DNA. We're also looking at the non-coding part of the DNA. We're looking at the whole DNA. We want to see which genes are there, where there might be changes, where there might not be any changes. The whole DNA is interesting for us. The pathological changes, but also the healthy parts of the DNA. This is something very, very, very important, especially when we're now moving towards personalized medicine. So personalized and preventive medicine. So in the last decades, the interest was focused more towards treating ill patients, um, people with certain diseases, certain syndromes. That's what the whole diagnostical tools and treatment plans were all, and doctoral visits were all focused about treating ill patients. But especially recently, and it seems to me like it started around also the COVID pandemic, where now the focus has changed or shifted more towards treating healthy patients. So treating healthy patients means something referred to as preventive medicine. So preventing the disease from happening in first place, preventing diabetes from happening. And so what can I do to decrease my risk of developing diabetes during sport every day, for example, diet changes? And what can I do to prevent or to reduce my cardiovascular risk, to prevent a stroke from happening in the first place, or to delay the happening of the stroke as long as possible? So this is something referred to as preventive medicine. There are many different levels to preventive medicine. Primary preventive medicine and secondary preventive medicine. We will not dive into that. So just to have an idea that when we're talking about multiomics today, and we will also talk a little bit later about personalized medicine. This is not something concern only about ill patients. It's also something that I would love to see, or I'm interested in also applying, but not just me, also many different doctors. That is something should be also applied to healthy patients, to allow also people who don't have a disease yet to understand what's happening in their body, and to help them prevent diseases from happening in the future. So now we talked about genomics, and we said that genomics is not just about looking at the pathological changes, at the mutations in the DNA, it's also about looking at the healthy parts of the DNA. We want to have a look at the whole DNA. So doing a whole genome sequencing, for example, is a type of genomics. Now, parts of the multiomic analysis. The multiomic analysis has different levels. If we look at the cell, the smallest part of the most important part of the cell, in my opinion, is the nucleus, for example. So we'll start at the nucleus, and then we will go higher to higher levels. So at the nucleus, we have the DNA. Part of the DNA is being transcribed. Then we are having RNA, and then these. When we go higher, then we will have proteomics, for example. So proteins, lipids, for example. So we started at the genomics. Inside the cell, inside the DNA, we have the genes, we have the DNA. So analyzing this DNA is a field called genomics. Now, analyzing parts of the DNA that are being transcribed, which means that parts of the DNA that are leading to changes inside the cell or outside the cell. So the part of the something like the active parts of the DNA. So the parts that are being transcribed are called transcriptomics, or the field that analyzes the part of the genes that are being transcribed is called transcriptomics. So the biologists here will think about RNA, which is the right thing. It's about RNA. So RNA are these very small parts that result from a gene inside the DNA that has been transcribed, and this RNA is a type of a messenger, it's a type of like a message being sent from the nucleus, from the DNA to the cell to do something. And the RNA is this message. So this also referred to as messenger DNA, as messenger RNA. But transcriptomics is not just about messenger RNA; it's about the whole RNA in the cell. So the whole RNA, this include messenger RNA, but it also include tRNA and also include non-coding RNAs. So it's about studying the whole RNA structure of the cell. Now I'll also talk about something that's also related to this, which is epigenomics. So epigenomics is a very interesting field, and I will certainly do an episode where I will talk about epigenomics in more detail. So epigenomics refers to studying the parts of the DNA that are active, parts of the genes that are active. And to understand this, you'll have to think about like if you have a gene, this does not mean that the gene is going to be transcribed. It does not mean that the gene will fulfill an action inside the cell or inside the body. And the epigenome talks about or describes this phenomenon that if you have a gene, whether this gene is being activated or being deactivated, whether it's fulfilling a function or not. And this depends on certain changes to the DNA structure. So the DNA, there's this theory about the double helix, that the DNA has a double helix, which is these two strands that are going inside each other, which is just a theory. It hasn't been proven yet, it's just a theory that works. And this double helix structure of the DNA, it's not just as simple as that, it also forms a lot more complicated structure that goes inside each other, and how the DNA is located inside the nucleus. It might be transcribable, which means that this RNA is being transcribed, and it might not be transcribable. And when certain changes happen to the DNA, like methylation, for example, adding a molecular structure to parts of the DNA, it makes it less transcribable. It makes it hard for the cell or for the nucleus to read this gene. So methylation is one example to this, by adding a methyl group to usually cysteine base inside the DNA. There are also certain other changes that might happen. Histone structure, for example, histone modifications, which are these proteins that are available around the DNA, which makes it harder for the cell to transcribe this genes or the DNA. It also depends on the chromatin structure inside the nucleus, that might be open or might not be open, which also makes it harder for the cell to transcribe the gene. So epigenomics is about studying these things, about studying the methylation, the chromatin structure, and the open chromatin profiling, histone structure and open chromatin profiling of the DNA. Now, one example I've recently read about epigenomics is, for example, that people with trauma, or in other words, trauma might lead to epigenetic changes inside the DNA. So this might lead to a deactivation of genes that is also transferable to their children. So you might have a gene, but because of trauma, this gene will be deactivated, and this deactivation of the gene will also be transcribed to the children of people with trauma. Now, as I said earlier, I will also make an episode where I will talk about this in more detail, because I think it's a very interesting field. But now, in the context of multiomics, epigenomics, which is part of it, is about studying these changes, exactly these changes. Which part of the genes are activated, which part are not, are deactivated or not accessible. So we talked about genomics, we talked about transcriptomics and epigenomics. Now we'll talk about proteomics. So proteomics is about studying the protein structure. Or remember when we talked about omics, it's about studying the totality of something. So proteomics is about studying the totality of the proteins in the cell, or if we're doing this on a tissue level, then the totality of the proteins inside the tissue. And you might think, okay, we studied transcriptomics, and usually these RNA, the transcripts are leading to building proteins. So we know that by having the transcripts, by having all the information about the transcripts inside the cell, we're able to know which proteins are being generated. Why would I study the protein under time, proteins, the cell under time? Well, because proteins undergo certain changes inside the cell. They might be phosphorylated, which means adding a phosphate to the protein. They might undergo acetylation. They might undergo certain molecular structures changes. And proteomics is about studying all of this. All these changes that all the proteins that are there and all the changes that are happening to them. Now, there's also another field called lipidomics, which is the same thing, but for lipids, to study the whole lipid structures inside the cell and what type of changes that are happening to the lipid structures inside the cell. Now, the next level is about metabolomics. So metabolomics is something that has been, it's a new emerging field inside multiomics, and the tendency now is to have like a separated field for multiomics that's only concerned about metabolomics, because metabolomics is so interesting, it's so effective. And metabolomics refers to studying the metabolites inside the cells. So let's go back to omics, the totality of something. So metabolomics is about studying the totality of the metabolites. And if we're doing this on a cellular level, then we are studying the totality of the metabolites inside the cell. And metabolites are very small structures. And metabolomics concerns or is concerned about studying metabolites that are less than 2000 Daltons. So this is these very small structures, these metabolites, they could be drug metabolites, for example. They could be amino acids, they could be sugars, they could be very small lipids, for example. It's concerned about these very small molecules inside the cell, or maybe also outside the cell. When we're referring to metabolomics, we're referring to these very small metabolites, usually under 2000 Daltons. So why is this field so interesting is that I've just said that metabolomics can analyze drug metabolites. So by being able to analyze drug metabolites, we are able to understand whether a drug is working and which dosage of the drug seems to be the most effective for this particular patient. So now we are more in the field of personalized medicine. So personalized medicine means providing a treatment, providing a way that is specially designed for this particular patient. And regarding to drugs, for example, let's think about ibuprofen. So ibuprofen is a widely known medication, and normally it's sold with a 400 milligram dosage. So this 400 milligram dosage might be very good for one patient, but might be very high for another patient, might be very low for a second patient. So using these metabolomics, for example, or in more general personalized medicine, we're able to know what is the best dosage to treat this patient. What's the best effective dosage to treat this patient? So this is on a very simple example. It doesn't really make a big difference whether you're taking 400 milligram or 800 milligram ibuprofen. But if we took this example and applied it to someone with cancer, for example, then it becomes a lot more interesting. It becomes about not just finding the right immunotherapy for a patient with cancer, but also about finding the right dosage of this immunotherapy to treat the patient. It's also about finding whether the immunotherapy works for this patient or whether we should be looking for something else. And it also reduces the side effects of this drug. If we're giving someone 320 milligrams and we realize that 320 milligrams are not leading to a better effect to this patient, he might be very well treated with 160 milligrams. Then we are able to not just reduce the dosage, the side effects, but we're also able to reduce the damages that might result from giving a higher, not useful dosage of a certain medication. So metabolomics is a very, very interesting field, and it has been growing, and since 2020, because this mass spectrometry, which is this most popular way to analyze metabolomics, has witnessed huge leaps, which allowed researchers to analyze and find these very small structures, not just on a tissue level, but also on a cellular level, one single cell. So this brings me to the next topic, which is single-cell multiomics. Single-cell multiomics. Single-cell multiomics is about doing multiomic analysis on a cellular level, one single cell. And if we are analyzing omics on one single cell, we might be, or it's very hard to integrate all these omics on one single cell. It's very hard to do genomics, transcriptomics, proteomics, metabolomics on a one single cell, because one method to do genomics might lead to changes in the cell that prevent us from doing metabolomics on the cell, or it might lead to certain protein changes inside the cell that which makes a proteomics not a very good idea. Doing proteomics is not a very good idea for this cell. And there are new methods to combine different type of omics on a one single cell level. So it's possible to do genomics and transcriptomics on one single cell. It's also possible to do genomics and epigenomics on one single cell. And there are continuously new technologies emerging that allows us to combine other types of omic analysis in a one single cell. Now, the other thing to talk about concerning multiomic analysis is about integrating all these data that we're generating. So the integration of multiomics. There are 3 different levels of multiomics integration, which is taking all this data that's being generated and making something useful of it, understanding what's happening with this inside the cell. So one type of omics integration is about vertical. It's called vertical integration, which means that the cell is the anchor, and we're looking at the epigenomics, the genomics, transcriptomics inside the cell. We're looking at the epigenomics, at the proteomics, metabolomics inside one single cell. So it's the same experiments, same tissue, same individual, same cell. We're integrating all these data together, and this is the perfect way to do a multiomic analysis. Also, one of the most difficult ways to do a multiomic analysis. As I said earlier, doing one type of analysis might lead to changes in the cell that will produce less significant data. Now, this is the vertical integration. It's also called match integration because we're taking the same cell, same tissue, same individual, same experiment, same time. And the other type of integration is horizontal integration, which means that we're analyzing just one omic, transcriptomics, for example, but we're analyzing it from different times or different experiments or different individuals. The same tissue type, but it might be other experiment, other time points. So we're now integrating these omics from different times, and we call it then the horizontal integration. The third type is mosaic integration or diagonal integration, which is integrating different types of omics from different tissues. So we're now talking about analyzing genomics, transcriptomics, proteomics, metabolomics from not just different experiments, different time points, but also different individuals. So this is one of the most difficult ways of integration because it's very hard to know what is the noise, what is the not significant data, and what's here the significant data that we're looking for.